Hi, and thanks so much for tuning in. My name is Kate Carbone. I'm a principal investigator at Seattle Children's Research Institute and an assistant professor at the University of Washington School of Medicine. Today, I'm going to be sharing some of my recent work, and the title of my talk is Intratumoral cDC1 T-cell clusters serve as sites of local co-stimulation to enhance CTL-mediated tumor rejection. What I'm going to walk you through today is how the spatial organization of immune cells inside tumors, and specifically the interactions between dendritic cells and cytotoxic T cells, can shape how well the immune system is able to respond to and ultimately reject tumors. Before we dive into the data, I want to just briefly go over the learning objectives for this talk so you have a sense of what I'm hoping to cover. After some introductory material for context, we'll talk about how the STAMP model enables real-time imaging of immune cell dynamics and how it lets us link immune spatial organization to tumor outcome. Second, we'll look at how cDC1 T cell clustering supports CD8 T cell function and contributes to tumor rejection. And third, we'll go through some of the key transcriptional and phenotypic features that distinguish cDC1s in tumors that regress versus those that don't. So this work is really about trying to better understand how the tumor microenvironment regulates immune responses. And as part of the broader conversation that is happening here at the LabRoots Immuno-Oncology virtual event, I hope it highlights some of the ways that deeper insight into immune spatial organization can point us towards new strategies to make immunotherapy work better. So, since we have a broad audience for this talk, I'd like to start by just briefly grounding us in the cancer immunity cycle, which frames a lot of how we think about tumor immune evasion and the logic behind current immunotherapies. So, in a perfect immune response to cancer, tumor cell death leads to the release of tumor antigens. These are then captured by antigen-presenting cells, particularly dendritic cells, which then traffic through the lymphatic system to the tumor-draining lymph node. There, dendritic cells can prime naive T cells, activating them to proliferate and differentiate. And these effector T cells then travel through the bloodstream, extravasate at the tumor site, and mediate cytotoxic killing of tumor cells, releasing additional antigen and perpetuating the cycle in a feed-forward loop. Unfortunately, tumors rarely follow this perfect model, so many steps along the cycle can be disrupted in disease, and each step represents an opportunity for therapeutic intervention. So my own background is mostly in thinking about checkpoint blockade therapies, inhibitors that target PD-1, PD-L1, or CTLA-4, or more recently anti-LAG-3. Originally, these therapies were thought to work primarily by blocking inhibitory signals expressed by tumor cells to allow T cells to kill them more effectively. But increasingly, we now understand that checkpoint inhibitors also act by enhancing T cell APC interactions at the level of T cell priming in the lymph node and possibly within the tumor microenvironment itself. So the 2010s were a period of extraordinary momentum in immunotherapy. Cancer immunotherapy was named Breakthrough of the Year by Science Magazine. It was recognized with a Nobel Prize in Physiology and Medicine. And, at the end of the decade, it was widely hailed as the decade of immunotherapy by major cancer research organizations. And advances during this time opened entirely new possibilities for treating cancers that were once considered untreatable. However, as we move into the 2020s, and now, in the midst of the 2020s, we find ourselves at a turning point. Many of the tumor types that are most inherently suited to immunotherapy, so those with high tumor mutational burden and pre-existing T cell infiltration, have already seen broad adoption of checkpoint blockade as standard of care. And as a result, although immunotherapy has delivered remarkable and lasting benefits for certain patient populations, the pace of new breakthroughs for additional tumor types has slowed. So even in our best case scenarios, and here I'm showing two examples on the slide, desmoplastic melanoma and non-small cell lung cancer, a significant proportion of patients still do not respond to treatment. So both of these cancers have high tumor mutational burden and pre-existing T cell infiltration, which are features that we typically associate with better responses to immunotherapy. And indeed, desmoplastic melanoma shows relatively high response rates. But even in non-small cell lung cancer, despite checking all the boxes we would expect for an immunotherapy responsive tumor, the outcomes are still, not great. And of course, many tumors don't even start with this advantage. So not all tumors are infiltrated by T cells at all. Across cancer types, we recognize three broad immunotypes. So inflamed tumors, shown here, where T cells infiltrate throughout the tumor parenchyma. Excluded tumors, where T cells accumulate at the tumor margins but fail to penetrate to the tumor core. And desert tumors, where T cells are largely absent. So this here is data from the IMvigor210 trial of atezolizumab in urothelial cancer. But if we look here, so as you might expect, inflamed tumors tend to respond better to immunotherapy. And this is nicely summarized in data from a triple-negative breast cancer trial of low-dose chemoradiation and anti-PD-L1, where you can see patient response is stratified by T cell immunotype. And so tumors here classified as immune inflamed, you can see show significantly better responses, while excluded and desert, here labeled as ignored, respond much less favorably. And across many solid tumor types, these excluded and desert immunotypes are common, and we lack effective strategies to address them. So the situation is even more complex in pediatric solid tumors, which represents an entire category of disease largely unaddressed by current immunotherapies. So many pediatric solid tumors lack pre-existing T cell immunity altogether, which is one reason why checkpoint inhibitors have not been widely adopted in children. In addition, pediatric tumors often present with multiple microenvironmental barriers to immune activity. So, to name a few, metabolic suppression, such as high levels of adenosine or glycolysis, fibrotic stroma or poor vascularization, impeding immune cell infiltration, and abundant suppressive myeloid cells, which can further limit T cell function. Despite these obstacles, antigen-targeted strategies like CAR T cells and bispecific antibodies have demonstrated that it is possible to engage the immune system in pediatric cancers. The challenge now is to deepen our understanding of the tumor microenvironment and how it constrains or supports immune activity so that we can build more effective and durable immune-based treatments in these difficult contexts. So now as we think about the cancer immunity cycle, it has been revised to the 2023 version, which includes this sub-cycle, really looking at what's happening in the tumor microenvironment itself and what sort of factors lead to local accumulation and proliferation of T cells, interactions with other stromal and immune cells in the tumor microenvironment, and effector maintenance for promoting T cell production of cytokines and, in some instances, local proliferation. And so this brings us to dendritic cells. Dendritic cells are myeloid lineage antigen presenting cells specialized for environmental sensing and T cell activation. And although they share a common core function, dendritic cells are a heterogeneous family with diverse developmental origins and functional programs. So here you can see summarized quite a lot of work looking at the differentiation trajectory of dendritic cells and the various subtypes that have been identified. And I think it's important to note that all of these commonly attributed features that dendritic cells can exhibit have exceptions. And so no single definition fits all DCs. So today I'll focus on conventional type 1 dendritic cells, or cDC1s, which really embody the full functional identity of a dendritic cell, and it's the subset that is most essential for priming CD8 T cells and supporting antitumor immunity. So in mouse models where cDC1s are genetically ablated, naive T cells fail to be primed to tumor antigen, And even when you transfer pre-activated tumor-specific T cells, those T cells fail to mediate tumor rejection without cDC1s. So clearly, these dendritic cells are indispensable both at the priming stage and within the tumor site. What remains less clear is exactly what cDC1s are doing within the tumor microenvironment. So several hypotheses exist. Dendritic cells may recruit T cells via CXCL9 and 10. They may promote epigenetic reprogramming towards more effective effector or stem-like phenotypes. Or they may support local T cell proliferation or cytokine production through costimulatory signaling. However, most of the work studying these possibilities has relied on static histology or transcriptomics, offering only a snapshot rather than a dynamic view of the role of dendritic cells. So during my postdoc, we developed a system to address this gap, the STAMP model, which stands for Skin Tumor Array by Microporation. And in this system, we use an infrared laser to create a grid of micropores in the skin of the mouse ear. We then seed tumor cells into these pores and allow them to grow into microtumors that are perfectly suited for live imaging over time. So you can see we create these pores, then apply a solution of tumor cells in saline. These can also be treated with Matrigel, and after about 4 to 7 days, we can come back and image these tumor arrays. And this is an example of what these arrays actually look like, showing the implantation of tumor cells into these pores in the dermis of the skin of the mouse ear. So this approach has several advantages. First, it's high throughput. A single mouse ear can host about 25 tumors, allowing us to study hundreds of tumors in parallel. Second, it allows us to isolate the role of the local microenvironment because all tumors share a common draining lymph node. Third, and quite importantly, it enables longitudinal dynamic imaging. We can track tumor growth and immune infiltration over time. And finally, we can do high resolution of cellular interactions as well to really capture immune cell biology in situ. Using this system, we applied 3-color imaging to simultaneously track tumor cells, which we can detect using MTAG BFP2. So here we're using the KPP tumor cell line, which is derived from a mouse gem model of pancreatic cancer, looking at the ARF-INC P10 mutants genetic context. We use GFP expressing T cells in the background of an XCR1 CRE TD tomato, which is specifically labeling cDC1 dendritic cells, which we also expand with the hematopoietic growth factor. And so with this approach, we can look at the role of dendritic cells in the tumor site and how they're interacting with T cells and with tumor cells. And we can do high resolution imaging to specifically look at the T cells and dendritic cells interacting within the tumor. And so this here is an example of one tumor lesion showing the T cells that have infiltrated and the dendritic cells which are also present. So across hundreds of microtumors, we observed 3 major immunotypes. These doubly inflamed tumors with T cells and dendritic cells infiltrating together throughout the tumor lesion. We see T cell inflamed and dendritic cell excluded tumors where T cells are able to infiltrate the lesion, but dendritic cells are constrained at the periphery of the tumor, and doubly excluded tumors which have both T cells and dendritic cells constrained to the borders of the tumor. And strikingly, tumors with both T cells and dendritic cells present were much more likely to undergo rejection. So now if we focus in on the T cell inflamed, dendritic cell inflamed versus the T cell inflamed, dendritic cell excluded population, you can see that this doubly inflamed tumor type is more likely to become rejected. So here we're looking at the survival probability of individual tumor lesions. So one important question that often comes up when I have given this talk previously is if all tumors are implanted from the same cell line, why do they develop different immunotypes? Is this variability intrinsic to the tumor cells themselves? So we did explore this carefully in our original publication. We showed that there are no consistent transcriptional subtypes or genetic mutations that can explain immunotype differences before implantation. So the variability really arises after implantation and reflects local microenvironmental differences rather than pre-existing differences in the tumor cells themselves. And once established, these tumors can mimic many of the molecular hallmarks observed in human tumors of different immunotypes. So these include Wnt beta-catenin signaling, elevated in desert tumors, TGF-beta signatures in excluded tumors, and interferon signatures in inflamed tumors. So in other words, the STAMP model recapitulates key features of tumor immune evasion seen in patients and enables us to make comparisons between individual tumor lesions within one ear and one mouse. And we can visualize cytolytic activity in real time using a GCamp-based calcium reporter, where flashes of calcium influx in tumor cells are indicating when you have T-cell mediated attack and perforin pore foundation. So here, if we look at the flashing in these doubly inflamed tumors versus the flashing in our T-cell inflamed dendritic cell excluded, you can see that the doubly inflamed tumors exhibit the highest rate of cytolytic activity shown here by these sort of hot spots of calcium flashing. And if we then calculate a GCamp flashing index across many different tumors exhibiting these immunotypes, we can see that these doubly infiltrated tumors exhibit the highest rates of cytolytic T cell attack. So next, we wanted to look at the spatial distribution of T cells and dendritic cells by histology to try to understand how our spatial immunotypes related to the histological observation of dendritic cell clustering with T cells in some clinical instances. And so we can see by looking in H&E and a multiplex IF staining for cDC1, CD4 T cells, and CD8 T cells, we can observe tumors that have exclusion of dendritic cells and T cells, so there's not many immune cells at all within the tumor boundary. We can see instances where we have T cell infiltration into the core of the tumor, but here in pink, you can see that all of our CLEC9A expressing cDC1 dendritic cells are at the periphery of the lesion. And we can also see instances where we have co-infiltration of T cells and dendritic cells. And what we noticed was the T cells and dendritic cells in doubly inflamed tumors tended to form dense multicellular clusters consistent with structures previously associated with effective anti-tumor immunity in other static histology studies. And this is in comparison to sort of unclustered cells as we might observe them more uniformly distributed, for example, in the stroma of a doubly excluded tumor. And we can score the abundance of these intratumoral clusters and see that in our co-inflamed tumors, about 70% of tumors or more are exhibiting this clustered behavior. So we think when we're looking at our immunotypes, we're really also understanding these interacting clusters that are happening on the micro scale. So to better understand what are the cellular and molecular hallmarks that support better rejection in these T cell infiltrated, dendritic cell infiltrated tumors, we sorted cells from pooled tumor biopsies for flow cytometry and RNA sequencing. And so looking at the genes that are enriched in dendritic cells with each of these different immunotypes, we found that cDC1s sorted from doubly inflamed tumors expressed high levels of costimulatory molecules like CD86 and ICOS ligand. They lacked a strong migratory gene expression. For example, looking here at CCR7 and Fashion1, which was really highly expressed in our T cell infiltrated tumors that lacked dendritic cells or had dendritic cells excluded. Um, and finally, when we looked at the gene expression of cDC1 dendritic cells in T cell excluded and dendritic cell excluded tumors, we saw really more of an immature dendritic cell type phenotype with a number of inflammatory genes like IL-1 beta and TGF beta, but low expression of sort of classical migration signature or co-stimulation signature genes. And when we look by flow cytometry, this result was largely supported. So these are very challenging experiments to do with such small tumors and with such a rare cell type. But we can see that our T cell inflamed, dendritic cell inflamed population, shown in green, had the highest expression of CD86, MHC class 2, CXCL9, and IL-12P40, suggesting really a T cell supporting phenotype of those dendritic cells. And by contrast, if we look at markers that were upregulated in our T cell infiltrated dendritic cell excluded tumors, we see more of this migratory or MREG type signature with high expression of CCR7 and other markers that have been seen in other studies, including CD200, PD-L1, CD40. So next we looked at sorted T cells from these pooled microtumors. And importantly, when we looked at these T cell infiltrated, dendritic cell infiltrated tumors, we saw this really very strong signature of a CD8 effector or antitumor CD8 phenotype. So high expression of molecules like CD8, TCR downstream genes like ZAP70, LAT, LCK, some genes that have been shown as hallmarks of antitumor T cells, including CCL5 and CD226, as well as markers of sort of early activation, including PD-1, SLAMF6, and antigen experience or tissue residency, including CX3CR1. Now, in the T cell infiltrated dendritic cell excluded tumors or the T cell excluded and dendritic cell excluded tumors, the picture was a little bit more complex because we see that there's much higher expression of CD4 in these samples. And so because now we're really looking at signatures that are coming from pooled CD8 and CD4 T cells, it's a bit hard to tease out, but we can see that there's sort of a mixed effector type CD4 signature in these T cell infiltrated dendritic cell excluded samples. So here we can see markers of antigen experience like CD44 and TIM3, effectors like TNF and interferon gamma, as well as sort of a suggestion that these are kind of a mixed lineage, Th1, Th2, Th17 in terms of the transcription factors that they're expressing. When we then look at our T cell excluded and dendritic cell excluded, it looks like they're really expressing sort of a Treg or TR1 type gene signature quite strongly. And so we can see a lot of hallmark genes like CCR8, TGF beta, the sort of co-inhibitory receptors that we think of as canonical for regulatory T cells. And so while we can't say whether these are explicitly coming from Tregs or, possibly these markers are also indicative of a terminally differentiated or exhausted CD8 phenotype, they are generally strongly associated with, a, a less good effector or an immune repressed state of T cells. And when we look by flow cytometry, we can see that in fact, Tregs are quite high in our T cell excluded, dendritic cell excluded population, and we have more CD8s in our dendritic cell infiltrated, T cell infiltrated population. When we then specifically look at CD8 T cells, we can see some signals of this sort of better effector CD8 with high CD226, high PD1, suggesting these are activated, and sort of an intermediate TCF1 and tox with both expressed, in contrast to our T cell excluded, dendritic cell excluded population, where we see really low TCF1 and high tox, suggesting a less functional or more terminally differentiated CD8 effector. So, given these findings, we asked whether we could manipulate the tumor microenvironment to experimentally favor rejection. And we did this using doxycycline-inducible constructs, where we can engineer tumors to express either CXCL9, which is a T cell recruiting chemokine, XCL1, which is a chemokine that's very specific for cDC1 dendritic cells, or CCL5, which is a broader myeloid attracting chemokine, which also acts on cDC1 dendritic cells. And so we can use this really nice experimental design where we have expression of our gene cassette in an experimental tumor cell line implanted into the left ear. And we have sort of an empty vector fluorescent protein expressing control construct that we express in the we implant in the contralateral ear. And so using the system and comparing the tumor rejection rates in the left to the right ear, we found that XCL1 expression really robustly increased tumor rejection, actually even about, almost twofold more effective than bringing in T cells directly using CXCL9. However, somewhat surprisingly, CCL5 did not appear to have a strong effect on tumor rejection. And if we look at the actual spatial immunotypes that are programmed by these different chemokine interventions, we can see here the control ear with a somewhat even distribution between our three different immunotypes. With CXCL9 expression, we see a large expansion of this population that has T cell infiltration without dendritic cell infiltration. And so it looks like it's really specifically acting on T cells without altering the broader localization of dendritic cells. In XCL1, by contrast, now we see a much larger population of this T cell infiltrated, dendritic cell infiltrated population, suggesting that we are acting on dendritic cells upstream of T cells. And what was quite surprising to us was that despite the fact that we see that CCL5 did not have an effect on tumor growth, we do see that it is driving a more favorable immunotype distribution with a higher abundance of these T cell infiltrated, dendritic cell infiltrated tumors. And so this, I think, is a really interesting observation and is likely due to the sort of broader pleiotropic effects of CCL5, including recruiting other myeloid cells and potentially having tumor cell intrinsic effects where it's directly impacting tumor cell proliferation. Okay. So, altogether, these data really suggest that local co-stimulatory dendritic cells acting within tumors are critical partners in effective anti-tumor T cell responses. So, looking forward, there's several major questions that we're excited to explore. Thinking about mapping where co-stimulatory versus migratory cDC1s localize in tumors to determine whether they represent distinct populations or transitional states of maturation. To identify which intratumoral cDC1 functions, antigen presentation, co-stimulation, cytokine production, which of these are essential in specific tumor types or specific therapeutic contexts. And last, dissecting how XCL1 signaling promotes the spatial reprogramming of the tumor microenvironment and testing strategies for targeted therapeutic delivery of XCL1. So before closing, I want to thank my postdoc mentors, Ira Mellman and Moussion, my wonderful collaborators, Lupe Ortiz Munoz, Marcus Brown, and Chimo Pechwan, and also the entire cancer immunology department, shown here as it was while I was at Genentech. Without all of them, this work would not have been possible. So it really takes a village. And, I'm very new to Seattle Children's Research Institute. The lab officially started March 1st, 2025, but I'm very grateful to my new colleagues here at Seattle Children's who are helping me get my lab up and running. And I'm excited to continue building on these foundations. And I look forward to collaborating with many people in the future. And please don't hesitate to reach out if you do have questions about this talk or would like to speak further about the work. So with that, thank you very much for listening in. I hope you learned something, and I'm really glad to have this opportunity to share my work.